Phil gave you an overview of what some of the ideas and the concepts for a project was, I thought I would talk about sort of the ins and outs of actually doing the field work. So the first thing that was a bit different for us was working with boats in the Marsh, which neither of us had done before. And so another faculty member at LSU had this hull that he was going to get rid of, and we said, we'll take it and we'll buy a motor for it. And even though that motor is rated for the boat, as you can see, it's quite heavily weighted at the back. And so our first experience, I think, on the third day in was this boat swamped to about here and gave me a heart attack because we almost sank our brand new motor. And then we found out that this hull had actually been swamped about 4 or 5 times before that, and it was lined with wood between the two hulls, and so that wood was probably totally waterlogged, full of sand and who knows what else. Then we had to go out and buy a hull, and frankly I'd rather buy a house than a boat because I don't know that much about them, and that was one of our very first challenges, was sorting out the boat. As Phil showed you, we're kind of in the middle of nowhere here, so we really need a boat to get to where we're going to look at these Seaside Sparrows. And the other thing about working in this area is that we're very constrained by the weather, right? We're not going out when the waves are big, we're not going out when it's windy or stormy. And so it really means that we have to plan our field work carefully. Okay, so this is our Seaside Sparrow here, as Phil showed you, and I thought I would start first by showing you how we catch these guys, our clever little rounding up technique, and hopefully this is all still working. So we set up a bunch of nets along the line, which you're going to see on the far right, and then we have a horseshoe of people around that net. The people on the sides are, well, I'm just going to turn this off, that's very distracting. The people on the sides are preventing any birds from escaping out the side, in theory. And then we have these long bamboo poles that we're kind of walking with and sweeping the top of the grass, so we scare these birds forward, moving towards the net. So these little white things across here with poles, those are all people that are moving in towards the nets, trying to round up our birds here. And we're trying to be sort of close enough together that they can't sneak out between people and between these little bamboo poles that we're hitting on the marshes. And shortly you're going to start seeing some little brown things zooming up from the grass and heading towards our nets. So normally we've got 3 or 4 nets set up across when we do this. So there's one there. Yep, there's one. There's some more. They're starting to come through, and here they come.
Are all those Seaside Sparrows?
No, there's quite a few Nelson sparrows.
Swamp a lot.
Batch, yeah.
Somebody get real excited.
Caught in our net.
One
got out. And then we go in and we take the birds out of the nets and we put them in bird bags and we take a bunch of measurements on them and band and blade them.
Scientist versus bird.
Yeah, usually the birds win, especially after they've been in the net a time or two. They get to be a bit sneakier about it. So we've got our birds, and here we've got our little setup here. So these are color bands for the legs so that we can tell apart different individuals. They have different combinations of color bands. So this is another thing you need to plan for. You may need to make a list of all the color combinations that are possible and then cross those out so that you don't give the same bird the same combination of color bands, right? The idea is for them to be distinct from each other. We take a blood sample, so that's what a lot of this gear is here. You can hold their wing open and there's a vein that comes across here, and you prick it with a needle and collect about 70 microliters of blood, which is a little drop. You put it in a tube which preserves it, and then that clots up and you let the birds go after they're banded in blood. And so the blood sample is in large part to look at the DNA and what population structure these birds are. It gives us some idea of how much the birds move around. If you have a really highly genetically structured population, so you have different populations that you can tell apart by their frequencies of the genes that are there, then that means that you don't have a lot of gene flow. If you have something where there's very little genetic structure, that means there's a high amount of gene flow. And we wanted to know this because when we're looking at our oiled and unoiled sites in Barataria Bay, you know, how much are these birds moving around? Are we going to see birds affected by oil that are dispersing somewhere else in the marsh? And so are we going to see the effects of the oil on a much larger scale from the areas that were actually oiled? Okay, so the nest searching, that's something that Phil mentioned. So this is Ryan, who is one of our field techs, and she was a fantastic nest searcher. We hired her because of her experience with boats, actually. She was really good at trailering boats and running them and knew all that kind of thing. And we hired 3 other field assistants that had vast experience with birds, right? She was the winner of finding the most nests that year. I think she found twice as many nests as the other 3 combined. I'm exaggerating a bit, but she was amazing. And she'd stand there and say, "Is that a nest?" Right at her feet, and there it was. She just had a great search image. So as you can see, it's kind of hard to see them, and they're very well disguised. And often what you're doing is you're watching the behavior of the birds, right? So if a bird is flying back to a particular spot and then disappearing there and never coming back out of the vegetation for a few minutes, you can go in and really keep a fix on where that bird went in and see if you find a nest there. The other thing you can do, which is a little bit easier, is when they have chicks, you can watch them carrying food into their chicks. So if you see a bird that's carrying food, you know it's got babies out there somewhere, so you want to keep a close eye on them and see where they're going. And luckily these birds aren't too cagey, they'll let you watch them do that. There are other birds. I was working on a grassland bird once that was carrying a big juicy grub. I knew it was going towards the nest. I was about, I don't know, it must have been 250 feet away from the bird and behind a boulder watching it, you know, so just my binoculars were over it. But it saw me and it would not go back to the nest after 15 minutes. It just ate this grub and then went off to find more food. Luckily these birds are much easier that way. They're not as scared. And they're also not as scared. I don't know if you've noticed when you come into the marsh, when you come up in your boat, all the birds will start popping up in the grasses to come and see what you're about. They're very curious. So it makes for an easy species to work with. Okay, and then here's a nest with the babies in. What we're doing to look at the productivity of these nests is we have to check them every 3 days. If we miss days, then we're not sure whether the nest got eaten up by a predator or whether those little chicks fledged, right? So we really need to check those nests every 3 days, which makes it tricky because in the height of summer, when you've got, you know, 50 nests and they've all got different times and your checks are all on different days, and then if you get a bad weather day, you can't get out to check some of these nests that you need to see. That's why we have some of those unknown fates on those nest charts, because we weren't able to get back in time before the chicks fledged or depredated, for instance. So it kind of adds an extra level of difficulty working in that area because the weather prevents us from getting out sometimes to when we really need to be out there. Okay, so the other thing we do, in these massive sets of data sheets we have, is we want to list on here how much time we spent netting on particular sites, how much time we spent searching for nests, and what our nest checks were here. Okay, so the reason we want to standardize the search time is because if we find 50 nests on a site, is that because we spent 100 hours there as compared to another site where we only spent 10? And so we really need to keep close track of this. You know, we've got four field assistants each year who are out in the field, and they've got all these data sheets they need to keep track of, you know, how old the chicks are, how old the eggs are, when was the last check, how long did you spend on each one of these sites. So as you can see, the amount of work starts piling up for these guys, right? This is an example of one of our data sheets for keeping track of the nests. So we want to know who the male is, so the color bands are how we would ID them. So the color bands on the right leg for the arm, the color bands on the left leg. Did we get a blood sample from this guy? Same thing for the female, all those details about her. Which plot we're on, the dates, when we saw our first egg, when we last checked. And then for a while we're using these little eye button devices, which are little probes that you can put in the nest, and they record temperature and time. And so when a bird is incubating on the nest, you have a higher temperature than when the bird's off the nest, right? So one of the things it helps you do is look at the incubation bouts of the birds, but it also helps you know when a nest failed. If you have a nice cycle of temperatures going up and down like this with the incubation bouts, and then all of a sudden you get a flat line at the same temperature, if the birds abandon the nest, then those temperatures are going to go down. So that's one way that you can look at nest failures and also how they're incubating. The other thing I want to mention is that obviously because these birds are so hard to get to, we don't know a whole lot about them, and one of the things we don't know is what their breeding system is like. So do we have monogamous males and females where all of the chicks in the nest belong to the males and the females that are feeding those chicks? Or do we have females that mate with multiple males, and so you might have a clutch of eggs that have a whole bunch of different fathers in them? We don't know that. That's why we want to get blood samples from these family groups, is to be able to figure that out. We can do that genetically. And another reason why we're taking blood, it will help us sort out what the social groups of these birds are. And the other thing that's a bit funny about them is how close some of them will put their nests to each other. I think we had a couple of nests that were only a couple of meters apart, right? 2 different pairs. And that's a bit unusual for birds. Normally they have much stronger territorial boundaries where they won't, you know, they won't have a neighboring nest.
What's the difference in
in
the color of the male and the females? The females are all brown, maybe?
No, they're the same.
It's hard to.
Yeah. So you can sex them in the field by when they're breeding. The females have those big brood patches, so they have that bare patch of skin on their stomach to help them heat the eggs. And the males have a cloacal protuberance, right? So their cloaca is swollen with sperm. We can also check that genetically too. There are markers that you can use to sex the males and females from each other. So
You don't take blood from the chicks after the
yeah,
day.
we do. We take them at a time sort of before they fledge, but not quite. You want to be sort of about 3 days before they fledge, because you don't want to prematurely cause them to fledge, but you don't want them to be overly young either. Yeah. Yeah, so time
So
of age.
have y'all found out whether the male, female, in the clutch with the DNA, if they're all?
If we've been, no, not yet. We're still figuring that out, and the reason is because we've had so few successful nests that have made it all the way to the time when we complete those chicks, those nestlings that are in the nest. So I think in the first year we had 10 nests or something like that, right? The second year, another 10 nests. I think we're up around 25 nests now, and just in terms of your ability statistically to say what's going on, you kind of need a bigger sample size than what we have. But we're really hoping that after this summer, we're gonna figure this out. Yeah. Okay, so the rats. Phil's showing you this picture. This is our rat person. She has a lot of experience with rats. As you can see, she's holding one of these Marsh race rats. These are quite little, cute rats. You can call a rat cute. They're kind of like a lab mouse size. They're about 30 grams or so. And here with the rats, instead of putting bands on their legs like you would with the bird, what you have instead is a little ear tag with a number on it. So those little houseboat traps that Phil was showing you, we can put those out, you catch the rats, you tag them, you've got their number, and then the next time you put the the traps out, you can see whether you're recapturing the same rats or different rats that are unmarked. And what that lets you do is estimate the population size based on the capture mark and capture. So you can try to look at differences in the population size between the oil and the unoiled areas by doing that. Yeah, and these guys, as we were saying, they're quite aquatic little guys. They swim really well. And here's one. Oops. Went down, who's been released afterwards. So that's what they look like when they're crawling through or running through the vegetation. Yeah, so that's basically how it works. So our 4 field assistants every year have a lot of work, and they have to be quite intrepid people because they're out usually, I think they leave at about 3:30 in the morning or something like that. They're trying to catch those early hours so that the birds and the rats don't get overheated when we're handling them, and so that they can come back at around 1 o'clock in the afternoon before they get overheated too, right? And there's a ton of different kinds of insects out there. You know, there's waves of midges first thing in the morning that'll eat you alive, and then the mosquitoes come in, and then the horseflies after that. And so that's why they're always wearing those bandanas and their hats. They're, you know, they just get punched by that. And so, yeah, they put a lot of work into it, these field assistants, when they go out there, and it's for many months on end too. I think they're out there for about 3.5 months in total. And the days they get off, we want them to be days that they can't go out on weather days. So if you get a string of good weather, then they're out there every day, and they take their days off when it's raining or the wind's too high or something like that, so that we can try and maximize the data that we're getting out there. So it's a lot of hard work, and even though sometimes you look at those graphs at the number of nests that we're finding, and you think, well, there's only 30 nests for 1 year on those unoiled plots, for instance. That's a ton of work. Finding those nests in the first place is really hard, you know, and then following them every 3 days, trying to keep tracking them requires a lot of work too. So that's some of the stuff that's involved with what we're doing. We're also looking at some of the trophic levels, like Mert mentioned. Linda Hooper-Boy is looking at insects. She's noticed differences in the insect assemblages on different sites, you know, between oiled and unoiled. So we're going to look at the diet of the seaside sparrows as well to see if those changes in insect abundance are moving up the food web into the seaside sparrows. And then I think part of our primary interest in getting into the rice rats was, first of all, we're the only consortium that I'm aware of that's looking at any species of bird at all. And I don't think anybody's even looking at any other vertebrates, are they? So we wanted to include these marsh rice rats as another level of the food web in here. And the other thing that I will mention too is that this marsh area is a really important area to study. That open ocean area where a lot of the dispersants were used, people aren't finding oil in the water column anymore out there. We're still finding oil in the marsh, so I think those effects in the marsh are going to be much longer lasting potentially than in the open sea. Any questions about any of this stuff? Yep.
Yeah, the birds stay right on the margin, or they move in throughout the salt marsh?
Yeah,
into the
they're
grass?
throughout the salt marsh. Yeah. Yeah, they're pretty high density, I would say.
So your study is the margin and inner marshes also?
Yeah, our study plots go, because we're looking at the effects of oiled and unoiled sites, and the oil didn't necessarily make it really far back into the marsh. We were trying to keep the areas consistent between the two. So our plots are 50 meters deep, so from the edge inwards 50 meters, so that's about 80 feet, right? And then along the edge it's 100 meters, right?
She's Canadian, more like 150 feet.
I was surprised you were using meters, actually.
Yes.
Way to promote science, Phil.
What made y'all choose Seaside Sparrow versus any other type of sparrow?
Well, the seaside sparrows are really abundant, so it's something that you can get a statistically robust sample size for. If you don't have enough birds, then it's really hard to draw conclusions that people are going to accept. And there's not really that many other species of birds out there. There's clapper-rails, red-winged blackbirds, grackles. Yeah, but the seasides are at quite a high population density, and there's a lot of them. And also, they're tied to the salt marsh. They don't live anywhere else, right? Like the red-winged blackbirds do, for instance. Yeah. And it's the same with the marsh rice rats. They're abundant. They're around. There's other things out there like raccoons, but you just don't catch them at those levels. And why probably Phil hasn't expanded us into snakes is trying to find like a houseboat trap to catch us.
An incubating bird. So in this case, this is an ant bird, an ovenbird from Brazil, but the same principle, and that is that you see this kind of loose tissue with these sort of creases in it, and then obvious blood vessels, and it all has kind of a soft appearance. So that's a bird that is incubating eggs. If it weren't incubating eggs, this would be smoothed across here. It wouldn't have these ridges. It wouldn't be vascularized like this. It wouldn't be this rich red. It would be kind of a duller pink. So it's a pretty good clue that a bird is on eggs when you see something that looks like that. So anyway, that was convenient, so I figured I'd show it to you.
Do you have a picture of the, oh, sorry.
Sorry, do they remove the feathers to accomplish that? Or?
They fall out.
They fall
That's
out.
associated with a whole bunch of hormonal changes associated with breeding.
I think canaries do that too.
Yeah, most birds do that. They have.
Canaries.
Pretty much any bird that incubates. Yeah.
Yeah.
So here's one. This one's in the Carolina wren. So that's not as fancy as the one we just looked at. You can really see the blood vessels the way you
Do you have a picture of the Seaside Sparrows' eggs, or?
Yeah, we do.
Sorry.
I could probably put this link on and you could look at these if you wanted. There's a handful of eggs. So you can number them so you can keep track of which one's which. If any disappear, then you have an idea.
Now, after you handle the eggs, the bird's okay with that? I mean,
Yeah, despite what your mom told you.
Same with the chicks. I think it's sort of a way to keep people from messing with the offspring in the nest, but
Yeah.
birds don't care.
You
You
have
take
to-
stuff out and put it back in, they, yeah,
Be careful how you take it out to put it back the same way, or it doesn't matter?
no, that doesn't matter. You have to be careful not to break them,
Yeah.
which is the trickiest thing, but yeah, they don't.
Actually, the birds themselves, when they incubate, intentionally rotate the eggs, because if they sit too long, then the placenta attaches, to the membranes, and it doesn't work. So they're like people that do this commercially for chickens. Say they put them on like one of those things at the convenience store with the hot dogs. They have that for the eggs to just keep them rotated.
That's a pretty big size egg for a small bird. I mean,
Yeah, I've seen that.
that's gonna be
About
1/3 of the size of the bird, am I guessing wrong?
Well, the whole clutch would certainly be, pretty good chunk of the weight. These birds are, what are they, like about 40 grams?
No,
Oh,
18 grams.
18.
That's not bad.
Is that their average clutch size, more?
3 to 4.
3 or 4?
Yeah, no, it's a huge investment by the parents definitely to lay eggs that big. But this, I wouldn't say this is a huge egg for a classroom there, but that size, for a group that size.
Or what time of year would, soon,
Yeah, our field assistants are gonna be heading out March 15th-ish, and do we have any nests this early on? Probably not.
I think we had some.
A couple started.
Yeah.
So not the majority of birds will have started yet, but there'll be a few early birds that will get going. And then they get really busy more into April, May.
Do they reuse nests?
Not that we've seen, right? The weird thing about them is that we find a ton of nests that have either been eaten up before we find any of the nests with eggs in them, or else the birds are maybe constructing practice nests before they, make a real one the way some birds do. We have a lot of nests that we never see anything in, and we're not sure what that's about. Now, last year we tried to nail down the predators a bit better by putting nest cameras on a bunch of nests. So what it does is it's a motion detector, and it'll take a picture when something's moving around the nest. The only thing we got was a raccoon, and it didn't take the eggs, if you can believe it. It just passed them by, yeah. And also one of the reasons we were interested in marsh rice rats is because they're potentially a big predator of seaside sparrow eggs, but we've never caught them in the act doing that. So there's lots to find out about both of these species, really. Yeah. And then, so we're gonna go out after lunch and try and catch some birds. We've got some mist nets and try this herding technique. And we probably won't get as many birds as there because the grass is quite short there still. But we'll definitely give it a go.
Right, yeah, disclaimer about the movie, right?
Yeah, that's right.
It wasn't doctored, but it's not gonna be like that.